All right, welcome. I wanted to make a short video just kind of describing some of the basics of the coronavirus mechanisms and how it works, just a little for your information. This isn't necessarily something that I'm going to specifically ask for on the test. However, the stuff is so related to what we're learning that some of it might appear on the test. And so I just think it's really coincidental that we're studying about RNA and protein synthesis, and actually two of my classes. So in biochemistry, we're really going into depth about protein structure, and with biology, we're studying just protein synthesis and how that happens in the cell through transcription and translation via the ribosome. And so this video I'll probably send to both classes. One will maybe know more than the other, but so I found a really good picture online that I think is going to be one of the best ways to kind of look at this. So follow along with me here. Let's see. Yeah, follow me down the tunnel of doom there. No, sorry, let me close that browser out then so you don't see that. Most of you are probably familiar with the images that have been shown online, whether they're artistic drawings or whether they're actual electron microscope images. I'm very fascinated with the electron microscope images that have been color enhanced, obviously, that show the structure of the coronavirus. Corona, means like a crown. And so the structure of a coronavirus has this crown-like shape. It almost looks like a sun. That's why sometimes we refer to the sun as a corona, having a corona. And so all of these things, what are those red things on the outside of that? I'm going to be careful here and not say the word cell. I've heard people say this is like a cell. The first thing you should know is that this is not a living organism. A coronavirus, as with all viruses, are not technically living because they don't meet the requirements to be a living organism of metabolizing food and having the machinery to do all the functions. In fact, that's what viruses are all about, is hijacking your machinery to do the work. And so let's look at how that happens. So these again just are pictures of coronaviruses showing the proteins that are essentially on the outside of this virus. So really all a virus is, especially this one, is just a protein coat. It's a protein coat with all of these different types of proteins on the outside. And in the inside, I don't know if there's any pictures here of that. In the inside, here you go, you have some RNA, and that's it. The problem is there's nothing to read that RNA. There's no nucleus to hold any kind of DNA. This is now some viruses have DNA. This is an RNA virus. It just has RNA, but nothing to read it. No other organelles, no mitochondria to take in sugar and process that for energy. So it can't make its own ATP. It can't do any cellular functions. It's not living. It can't reproduce because it doesn't have any way to make energy, and so it's helpless on its own. But it does have RNA. It does have RNA, which codes for the same proteins that make up this outside of the virus. And in essence, it can still replicate itself. And that's exactly what it does, is it uses a cell to do that. So let me find the image. This is a good one. Here we go. You essentially have all these proteins on the outside, and then the inside you have some RNA. And so here's what happens. These proteins, and if you've done any kind of listening or watching or have gotten into this in any way, you've probably heard people talk about this special spike protein. This spike protein on the outside of the coronavirus allows it to attach to this receptor here. I'm not going to go super deep into names and all that of what the receptor is and all this, but the point is, this is like a lock and key mechanism. This spike protein is like the key that fits into this lock, which is the receptor. And when that happens, the cell is going to engulf it. And this picture shows that best. This picture shows that best. When the virus attaches like a lock and key there to that receptor, then the plasma membrane will fuse together around this and make a little capsule and then bring it in. Right, so it first forms this little capsule. It's called endocytosis. That's the way that cells engulf stuff without just opening their cell membrane and letting all their guts spill out. So they do it in a nice way that stays sealed. And so it's going to engulf the virus. Okay, what happens then? Well, once the virus is inside, it'll open up, and guess what? This RNA will be, first of all, this is the interesting thing to note, it'll actually be copied a bunch of times. So there's actually an RNA-dependent RNA polymerase. Now, normally RNA polymerase reads DNA and then makes transcripts or photocopies, if you will, so that the DNA can stay intact, and then the RNA can get read and translated. But in this case, there's actually an RNA-dependent RNA polymerase that can just simply copy this RNA strand over and over and over. So we're going to get a bunch of photocopies of this RNA. All right, so we're not done yet. We still haven't finished the mechanisms here. Once that RNA is copied, then it'll be, remember that's called transcription or just replication. So we're going to have a whole bunch of these, and then they get translated. So check it out. Let's say this gene here, whatever, will get translated to that protein, this S protein. Maybe this gene here, which might be this little section, will code for some other type of protein. And then this section will code for this protein, and this maybe another section here will get translated by the ribosome into this protein. And so we have all these proteins here. We have like a laundry list of proteins that get made by the translation of this RNA. This is exactly what we're studying, right? This RNA will be translated by the ribosome, and we'll make a long amino acid chain that is called a polypeptide, and that'll go through some folding and twisting and three-dimensional structural rearranging to end up with these proteins, right? And so you'll have these individual proteins. They all serve different purposes. Well, a lot of the proteins are actually serve the purpose of being on the outside of this corona. Like, look, this is a spike protein. Well, we just made another spike protein down here via this RNA. So the RNA inside of the coronavirus codes for making the proteins that give it structure. Remember, that structure is what allowed it to get into the cell in the first place. So after these proteins are made, they'll go to the endoplasmic reticulum where they'll attach, and then that'll bud off or fuse off and then fuse, and then RNA, these copies of RNA will be put inside as well, and then it'll go to the Golgi apparatus where it'll be modified and sorted and packaged and sent out into vesicles, and then you essentially have your duplicated coronavirus. And this will just happen over and over and over and over, and your cells, these cells are in your lungs, by the way. These cells, that's why you have such a horrible reaction in your lungs, because your lung cells are basically hijacked, and the ribosomes, which would normally be doing their other jobs and making other proteins, are too busy making these proteins and making more and more and more coronaviruses, and then they get sent out through exocytosis. So endocytosis brings it in, exocytosis sends it out, and guess what? This coronavirus is probably just going to head over to the neighboring cell for it to happen all over again. And before you know it, your lungs are overwhelmed because all those lung cells are doing this. And so this is interesting. This, hopefully, I think you guys can hopefully get an appreciation of how, I would say, easy this is to stop, but yet not quite so easy, right? The experts are the ones that are really having to go to work to do this, but from the couch perspective and basic level biology, you can see that these mechanisms, hey, if we can just stop this coronavirus from even getting in the cell, we would be good, right? In other words, if we can disable these keys, change the shape of these keys some way so they don't fit in this lock, or if we can put something else in this lock so that these keys can't go into the lock, then you won't have that key and lock situation, because once that happens, that'll allow the coronavirus to get into the cell. Another thing you could do is stop the translation of this RNA. So any of these RNAs that get copied, you could stop the translation of them, and then you won't get these proteins. You could also stop the copying of this RNA. So you could deactivate or disable this RNA-dependent polymerase. But again, these could have other side effects on the cells too that you may not want. And so it sounds easy, but it's not quite so easy, but it kind of is, right? And so if there's any way we can prevent these proteins from attaching to these receptors, then we're in pretty good shape. So that's just one of the ways, and there's other ways too. I haven't done all the research on what mechanisms they're coming up with that could essentially recognize these viruses and get rid of them or stop them from their work inside the cell. Once you start stopping the healthy cells from doing their work, then you're kind of in trouble, because you're you need these lung cells, right? This whole square here represents a lung cell. I call it a lung cell. It's probably another word for that. This starts with pneumo, right? All right, so I just wanted to hook you guys up with a little FYI about how the coronavirus works. Remember, it's not living, but it is simply just a protein capsule with some RNA inside of it. And so hopefully as you're learning about in biology, like I said, if it's RNA and how it can be translated into proteins, or whether you're in my biochemistry class learning about proteins necessarily, then hopefully you found that information useful. If you're in the biochemistry class, it'd be interesting to look at the actual amino acid sequences there, and the real structure of those proteins in terms of like the helixes and the sheets and the different intermolecular attractions that are going on there, and how you could maybe do something to get in the way of that or whatever. But okay, all right, enough of me. Bye.